conference, I think, starting from 2019, where he was in person, then 2021, when he was online, and now also online. Hopefully, next time he will be also in person. So, yeah, we have deep collaborations with Professor Wolf, so let's see what he will tell us about. And yes, please, you can start and share your screen.
Okay, thank you, Professor Sokolova. It's always an honor to participate. Actually, I was there in Moscow at the first RICCEM conference. Can you all hear me all right?
The first conference was in 2017.
Yeah, okay. So I will then start. So thank you for inviting me. I will share my screen. Can you see?
Yes, everything is okay. Thank you.
Okay. Yeah, sorry, I had the wrong link apparently before. So, right. So I have 25 minutes. Is that correct, or is it shorter now?
No, I think you can speak for 25 minutes and then 5 minutes for questions and answers.
All right. Thank you very much. So again, it's a pleasure to address you and to present this work to you. I regret I'm not there in person. As you can see from the title, I will talk about a virus, about pox virus. Somehow this does not, yes, okay, now it's working. All right, this is work by Dr. Chae-Kyung-hyun, who is now a professor at Kungsuk-Yong University in Korea. He's one of the well-funded universities in Korea. And this was published last year in Nature Communications. Dr. Chae-Kyung-hyun is here in red, so this was chiefly his achievement. And the rest of the people is my group at OIST. I'm going to talk about immature pox virus. It has a so-called pleomorphic or irregular non-icosahedral capsid. And I will just briefly introduce this genus of viruses and then show you three structures. And then there's also a tomogram and give you some of our scientific insights. So pox viruses have been known for a very long time. Even the Egyptians, apparently in sarcophaguses in the pyramids, they found remnants of pox virus. They were haunted by this virus. And as you know, the most famous one is smallpox, and it was eradicated in 1977. So pox viruses are large enveloped double-stranded DNA viruses. And the one I'm going to talk about and that was used in this study was vaccinia, and vaccinia is the most harmless one. Vaccinia also gives its name to vaccination because the milkmaids in France who milked cows that were infected with cowpox virus, they turned out to be resistant against variola, the actual dangerous human pathogen that killed many people in the world. And so this initiated vaccination, essentially the systematic cultivation of a strain that was attenuated and in the end could be administered as a vaccine to cause immunity in people. It was the first successful viral drug or the virus that was used for creating this kind of immunity. NCLDVs are nucleocytoplasmic large DNA viruses. So they have a very large capsid on the order of 200 to 400 nanometers. Compared to the coronavirus that causes COVID, and coronavirus is up to about 200 nanometers. You know, pox viruses are even larger. They also have many more surface glycoproteins, and they have 12 to 15, I think, glycoproteins that initiate membrane fusion. Compared to the spike protein on the coronavirus, which has only one, this is a much more complicated virus, and is therefore it's very difficult to find a successful strategy against it. So the best strategy is vaccination, but thankfully since 1977 this problem is gone. But who knows, it may come back eventually. Right, so I'm going to talk about the IV, the immature virion, and its scaffold is different to the NCLDVs that form icosahedral scaffolds. So the IV doesn't have an icosahedral scaffold, but it has a smooth, round sphere that is about 400 nanometers in diameter. And it's locally irregular. So the question is now, like, how does this form? This is just as a background, the poxvirus life cycle. These poxvirus virion particles on the left here, like, they form these characteristic dumbbell-shaped structures, and they get endocytosed by clathrin-mediated endocytosis. There's another entry pathway as well, and then once the DNA is released after uncoating, it's taken up into the nucleus, where initially, as a first gene product, these crescents form that then assemble into the IV particle here. And this is the focus of this study. This IV particle then undergoes a dramatic conformational change, forming essentially the mature virion that then gets released and goes on to infect additional hosts. Now, I'm showing you here an electron tomogram, and you can see the circular outline of IV particles assembled from in vitro expressed D13 protein. And it forms a honeycomb-like pattern, just like the wild-type IV that has been found in cells. We call it the honeycomb-like pattern because it looks like honeycombs, and I hope I can catch it here. Oops, pause. Let me go back a little bit. Yeah, so around here, so you see these hexagonal arrays of the D13 trimers, and in this cross section through the tomogram, you can get a first glimpse. And so the IV particles that we worked with, they were expressed in vitro. Now, when you put them into a thin ice layer, like what we do by single particle cryo-EM, we plunge them using a plunger after blotting, and then this results in a thin ice film that is on the order of 50 to 150 nanometers, something like that. So it's quite thin, and of course these particles cannot remain spherical, so they flatten into a plane, and then you have these breakages in between. They are not connected anymore everywhere because they want to form a sphere. But nevertheless, you can still see the honeycomb arrangements of the D13 trimer protein. Now, after doing 2D classification of patches from these images, you can see an enhanced clarity here that there are mostly hexagonal arrays of trimers, but also occasionally there are pentagonal arrays. And then there are, at the interfaces here, there are 4 and 3 and sometimes 2, and all sorts of different assembly states. Now, we of course, the goal was to determine the high resolution structure of D13. Unfortunately, when D13 was expressed in vitro, and then at a high salt condition where the particles were nicely separated, they unfortunately only showed top views. So when you see here the classes, there are only top views, no side views. And so for reconstructing images from projections, you want to have all orientations in 3 dimensions, ideally randomly distributed. So ultimately, Dr. Jae Kyung overcame this problem by using a graphene oxide support film. And you see this on the right side. The graphene oxide is a very thin film that is layered on top of the holy carbon film that we use usually for cryo-EM, and it's almost adds no background, but it adds the fact that the protein can lie down on it, and so they like to lie sideways on it, turns out. So this way we got many side views, as you can see here, and then also some oblique intermediate views and occasionally some top views. But by combining these two data sets together, we would then get a very nice data set that had initially, I think, almost, well, so here it says 200,000 particles, and I think this was already sorted out a bit, so probably 500,000 particles. And then after 3D classification, turns out that this class included threefold symmetry in position, then went to 2.2 or 2.3 angstroms resolution. And you can see here nicely an example of the electron density map, or the electron potential map, that shows nicely featured aromatics and side chains. One thing that was immediately obvious once first of all, when we looked at the map, there was a lower local resolution in these regions that are colored here. And then after fitting the model, so the atomic B factors were much higher in this, what turns out to be the N-terminal helix of D13, had a much higher B factor, temperature factor. So indicating that it wasn't very well coordinated and sort of rather floppily attached to the sides of this trimer. This shows now the reconstructed map, and you can see the nice features, like the alpha helices are clearly visible, the side chains are there. Now this looks a bit messy, but we'll go through it in slices. And especially when the model is docked, it's obvious that there are also water molecules that are clearly resolved at this resolution, and wasn't, you don't see the holes in the aromatic rings, but it was close. So it's a very rigidly assembled trimer, and there's 3 subunits in it. And wait, next slide. Here is a schematic. So 3 D13 molecules form this trimer, and it's so rigid because the N-terminus here invades, this arm invades the next trimer, and the red one goes over there, and then there's a yellow one goes across. So it is locked together and very stable. But yeah, here is that variable domain that we noticed early on. It's the N-terminal alpha helix, and it's shown here in close up. So this forms a so-called beta jelly roll fold, and the N-terminal alpha helix is at the periphery. And it's stabilized in a hydrophobic pocket, leucine, valine, and there's many hydrophobic residues there. And this was not visible in previous crystallographic maps. It was only visible in this cryo-EM map. Otherwise, the structure, even though it was actually even higher resolution than exocrystal structure, it didn't really reveal any novelties. So only novel part was really that this alpha helix was present, and it was locally lower resolution. Now, we wanted to know what happens, how the neighbors interact with these trimers. So it turns out that there was a subset of dimers of trimers in this very same preparation. This time, however, the graphene oxide technique wasn't enough, and we had to substitute additional side views by tilting the stage to 45 degrees. So we collected an entire single particle data set, total 750,000 particles, using a tilted stage, 45 degrees, always tilted, and then combined these data. And with that, we were able to get to 3.9 angstroms resolution for the dimer of the trimers. And although the density was slightly anisotropic, so it looks like a little bit elongated in one direction, in the Z direction, it was nevertheless possible to fit the atomic model, adopt the atomic model quite faithfully. And at that point, we were able to get insights into how the trimers interact with each other. And we can already get an idea that, is here. Yeah, I'm not gonna wait for this. Here you see a schematic that two dimers together, and they form interactions at the top between the yellow parts and then at the bottom between the red parts. And the key interaction is essentially this aromatic cation interaction, tyrosine 62 and arginine 498. So they are at blue and red at this interface here. However, what was remarkable was that the density for the N-terminal alpha helix was completely gone. So there was no density visible for it. That suggested that somehow the dimerization pushed away the N-terminal alpha helix. Or to take it differently, the displacement of the N-terminal alpha helix initiated the assembly. So to give a bit more credibility to this, we expanded this hypothesis. We expanded the symmetry linearly. And here you see, like if you expand it, you get essentially the curvature of the immature virium membrane. Also, when looking at this more closely, it turns out that the axes of the trimers, they were twisted by about 8 degrees with respect to each other. And it was this twist that by snapping together essentially at the top and the bottom that is generated by this interaction, that essentially implies curvature by design. So this symmetry expansion yields curvature. However, when you expand it in a circular fashion, then it doesn't close up. So here again, you see the linear expansion, and then in the next slide, circular ring-like expansion. That one has a gap here, so it has a seam, and it does not close up, suggesting that there is must be another mode of interaction that enables, a three-dimensional assembly that results ultimately in a spherical curvature rather than just a linear circular curvature. Now, this is kind of a key slide. So the one main experiment was that at low salt, using the wild type, one can induce the formation of these honeycomb-like precipitates. And this needs to be zoomed in a little more, but there are honeycomb structures in there. And the wild type has the helix, N-terminal helix present. Just, this is what we use to get our high resolution structure of D13. But then when J introduced by mutagenesis his tag on a longer spacer, apparently the introduction of charge into this hydrophobic pocket displaced that charged his tagged N-terminal helix and initiated the assembly into what looks exactly like the known D13 immature virions. So this introduction of a charge of the his tag on the N-terminus resulted in IV-like structures. And then finally, when this N-terminal helix was completely truncated, another polymorph was formed, this tubular structure. And in the tubular structure, in the reconstruction, neither of them, the N-terminal alpha helix, was present. So essentially, we concluded from this that the assembly of D13 requires the displacement of the N-terminal alpha helix, because here, where the charge pushes it out, or doesn't allow it to be in the hydrophobic pocket anymore, essentially that leads to disassembly. And there is a so-called a protein called A17 on the surface of the IV, that is a transmembrane protein. It's anchored to the lipid membrane, and A17 is known to recruit D13 to the membrane, and A13 is required for initiation of the assembly of the IV particle. So the hypothesis is essentially now that the N-terminal alpha helix gets displaced towards the lipid membrane and allows interaction of the D13 trimers with the membrane-bound A17, which is in purple. Right, it's indicated by these ellipses. So we don't have a structure of that complex. Yeah, so for completeness, here is the structure of the tubular array. So we used helical processing for this, and there were several different diameters, and it was a bit tricky to get the right helical symmetry, but ultimately we got it. And so here's a tomographic reconstruction of these tubes. There also has a nice honeycomb-shaped pattern. So it zooms, goes through the tomographic series. And then here's the reconstruction. It went to 7.3 angstroms resolution. The projections from our reconstruction and the 2D class averages, they were consistent, and the power spectra were also consistent, suggesting that we had a correct symmetry. And then this is the reconstruction. These hexagonal arrays are all formed of D13 trimers, and it's going to get colored in, so you will see where they are. I think so, yes, there they are. And so it turns out in this array, in the tubular array, because it is curved in cylindrically curved, there was another interaction motif. So we studied again the motifs of interactions between the trimer, and we found that there's a new one. So here the resolution could be extended a little bit more using local refinement in the program CryoSpark, and this worked very well. So within a focused mask, essentially, we were able to exploit additional symmetry that was not contained in the helical reconstruction alone, and push the resolution to 4.3 angstroms. And again, so here's the map, and you can see from this reconstruction that the map is cylindrically curved smoothly. And yeah, the resolution wasn't as high, but we were able to dock our structure into it. Now this is the schematic of the different interfaces. Essentially, when you go from a cylindrical to a spherical assembly. Okay, so maybe I have to back up here, because we didn't get the spherical assembly. We got the cylindrical assembly and all the previous ones. But it turns out that there were two motifs of interactions, indicated by a black star and white star. And they alter black, black, white, black, black, white. And that gives the cylindrical assembly. However, if you in the computer then alter this, and it goes no, this is incorrect. This is the top and the bottom of the cylindrical assembly. I apologize. Would be the next slide. If we alter this arrangement of assembly motifs, interaction motifs between the two dimers, from black, black, white, black, black, white to black, white, black, white, black, white, then we actually end up getting a spherically curved assembly that does not have a seam and that perfectly seals up. And this is the cylindrical one that forms the tube, and then this is the one that would form the spherical assembly. So I should say that we only have a tomogram of this at low resolution, but the single particle data essentially and the modeling here are very consistent to cause the fully curved spherical curvature. So here you see the comparison between the cylindrical and the spherical assembly, and the spherical assembly again was put together in silico. So we have experimental data only for the left one. Now, when you look at the charge distribution, essentially these D13 building blocks, they are charged in a way so that they snap together in the correct orientation, because they have complementary charges at their tops and at their bottom. So I'm sure I haven't done this, but I should probably 3D print them and put like little magnets in or something. So I bet if you would put them then in a bag and you would shake it, you would end up with fully assembled virions, just by means of the charge complementarity. So this is a very efficient mechanism of self-assembly. Now our reconstruction, this is the tomogram here of the fully assembled IV, is consistent, with the previous freeze fracture data. that is published before. So we are confident that this assembly is correct. Yes, now to summarize, essentially, our assembly hypothesis goes like this now, that, at first we have the 13 trimers that are loosely linked. It depends on the ionic strength, also in the buffer. Low ionic strength causes aggregation. There's different numbers of trimers that can form together and can come together. And then the displacement of the N-terminal helix of D13 to the viral membrane by A17 initiates the assembly by exposing the base-to-base interface. And then first starting with 2 trimers getting together, then the twist between these trimers then determine the local curvature. And this finally sort of expands into the honeycomb-like lattice. There is just sort of a note on the side that, Kasper, Don Kasper and Aaron Klug have in the 70s, put forward their universal theory of quasi-equivalence, where you have building blocks that, lead to the assembly of icosahedral assemblies, but also other assemblies by means of the triangulation number. And this has been very helpful for explaining the symmetries of many viruses. And it seems like our structure is an exception to this rule. Sort of, it has an underlying icosahedral symmetry. However, you don't usually find 5-fold axes. Icosahedral symmetry is 5, 3, 2, and these IV particles form round, smoothly curved assemblies that don't have the icosahedral symmetry. However, as I have shown, in some of these particles, before they're here, you can see there are also 5. So in principle, it's possible that 5 of them come together and form a 5-fold icosahedral vertex. However, the dynamic nature of this assembly likely requires a certain flexibility. And this is a unique feature of these NCLDVs and the IV of the vaccinia virus. Yes, and with this, I am come to the end, and I want to thank you for your attention. And we're happy to take any questions if there's still time.
Yes, thank you very much for being just in time, despite you unfortunately started a little bit late. So, colleagues, any questions? Yes, please.
Hello, Matthias. Thank you for the presentation.
Hi.
Actually, I do have 2 questions. One is kind of a technical. When you speak about the helical reconstruction, I think you mentioned that there were several different helices present. How did you distinguish between them? I mean, how did you reconstruct different symmetries? This is the first question. And the second one is about the native structure. Actually, as I understood, it is a little bit disordered. I mean, there are several partons present in the one virion. So is it possible to reconstruct these partons with some kind of the subtomogram averaging of the actual particles? Thank you.
Yeah, thank you. Those are both excellent questions. So the question, the first one about distinguishing the helical symmetry, the most obvious parameter is the diameter of the tube. So this is fairly easily done by 2D classification, and some of the results are shown here. But as you can see, some of them have a very different helical appearance. So it's indeed, there must be different assembly modes, and it probably depends on, how many asymmetric units are in one term. We essentially cracked this by picking the classes that had the best visible features. And here you can see those red ones are combined. Then, of course, determining the actual helical operators, meaning the rise and the rotation of the asymmetric unit, that you go from one projection to the next to recreate a 3D structure is not trivial and requires a bit of insight in Fourier analysis. But there are some papers out there, and nowadays also very helpful program called PyHi that allows you to plot the lattice. So we essentially use that and also the local helical refinement built into rely on to get to the refined helical parameter. But yeah, so the main distinguishing feature was the diameter of the helical tube. And so your second question was whether you could do subtomogram averaging within the reconstructed lattice by tomography, and absolutely yes. So in this paper, we didn't do this, but Jay actually tried it, and he couldn't get beyond the resolution of 7 angstroms that we got here with the helical one. But I suppose if we had enough of them, we could reconstruct also by subtomogram averaging to higher resolution. So yeah, no, very good questions, and I think it's absolutely possible. I'm not sure if that answers the question.
Thank you.
Yeah.
Any other questions? No more questions? So thank you again, Matthias, for a wonderful talk.
Thank you, Olga. I really regret I'm not there. So.
And I hope to see you again in the nearest future. Okay.
Okay.
So next, bye-bye.
Bye.
Next speaker is Valeria Samygina from National Research Center Kurchatov Institute, and she will speak a little bit about some other viruses. Пока у нас еще нету презентации, я хотела сказать, что мы собираемся выбрать лучшего молодого выступающего, спикера, да, который очно на нашей конференции делал презентации. Поэтому, пожалуйста, не разбегайтесь, во-первых. А во-вторых, попросите всех ваших знакомых прийти сюда, чтобы мы могли выбрать лучшего презентера. Но не только за сегодня, а за все 4 дня нашей конференции. Единственное, что я не могу точно сказать, кто у нас считается молодым презентером. То есть, видимо, до 35 лет. Поэтому, но если вас выберут, то вы будете считать, что вас считают очень молодым. Да, насколько выглядишь, настолько и... Угу. Прекрасно.
Раз, раз. Раз, два, три. Раз, 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 раз. Слышите меня? Раз, раз, раз. Нет, работает. Работает, да.
Куда его цеплять?
Сюда повыше.
Вот так вот, да?
Ну, так нормально.
Нормально?
Не очень. Слишком мягкий вверх.
Что делать? Вот так делать?
Ну,
Лучше?
попробуйте.
Раз, 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 раз.
Да, есть звук.
Слышно, да?
Да.
Все, тогда я пошла. У вас тут лихо вообще. Sorry for technical problems, and could I check this one? Okay. Good morning, dear colleagues. First of all, I want to say thank you organizers for inviting me to this nice conference. And today we are talking a bit about pipeline for characterization of inactivated tick-borne encephalitis virus samples for single particle structure determination. Structural virology and structural vaccinology is the attention-grabbing part of modern structural biology because emerging infection diseases will continue to threaten public health. And there is no specific treatment against some dangerous pathogens still, and there is no effective vaccine against certain pathogens, or there is necessity to improve its effectiveness. And case of COVID-19 is unforgettable, and this virus came to every family, but except this dangerous virus, we have a lot of other ones. And knowledge about detailed virus structure and dynamic can broaden our arsenal of weapons against those old enemies of humans and animals. If we know 3D structure, we can know exactly structure and position of viral epitopes. We can create new vaccines or develop engineered ones. We can find new inhibitors of antigens, test systems. And one of pathogen for which there is no universal vaccine, for example, is dengue virus. It's endemic in 100 countries, and new regions are reporting cases of this virus every year. Dengue virus belongs to flavivirus family. These flaviviruses are now globally distributed and infected up to 400 million people annually. There is more than 50 members in flavivirus family, including tick-borne encephalitis virus. Tick-borne encephalitis is a viral disease that has a substantial epidemiological meaning for Europe and Russia. It's about 2,060 cases per year in Europe, for example. And clinical manifestations of TBE vary from uncomplicated fevers to potentially lethal encephalitis or meningoencephalitis. And TBE is transmitted by ticks and becomes more spread due to climate changing and global migrations. Here you can see the screenshots from news channels. One screen is a news from Massachusetts, USA, and it's about ticks that activated by warm days during winter. And every winter from year to year have more and more warm days. And second channel reported us news about cases of TBE in New England. This disease is very untypical for this region. What about structure of this virus? It's enveloped virus, single-stranded RNA. The virion has diameter about 50 nanometers and envelope formed by protein E dimers in a herringbone-like arrangement. M protein placed just under the E protein, and membrane parts of these proteins are immersed into membrane, and C protein forms nucleocapsid with RNA. All enveloped viruses are structurally heterogeneous, and you always will have in your sample different forms: mature form, immature form, broken particles, and half mature, and so on. So, this is illustration of this heterogeneity. This is cryo image of tick-borne encephalitis virus Sophin strain. And you can clearly see the different type of particles. This is mature particle, this is immature, a broken one, and this also broken one, and this half mature. Okay, so, and heterogeneity of these virus particles limits us in the choice of method which we can use for structure determination. And of course, first choice method is cryo-EM, single particle analysis, for which we need a frozen sample. But also there is a younger structural biology method called single particle imaging using free electron lasers. This method based on diffraction before destruction principle. You have stream of very concentrated particles. You have to deliver it under the bright beam, detect the diffraction patterns from single particles, collect millions of images and processes, and luckily you will have structure. But for SPI experiments, virus particles are big enough to see diffraction. This is modern because, for example, attempts to see diffraction from ribosome was unsuccessful today. So viruses is a very good model object for method development itself, because this method is still under development, every step of this method. If we come back to cryo-EM, this story of structural studies of flaviviruses is very long. So first structure was obtained at 2002, 20 years ago. It was dengue virus. Structure was solved in 24 angstrom, and you can see the progress of the studies, because recently in 2021, mature structure of Dengue virus was solved in 2.6 angstrom resolution, and you can find about 100 cryo structures in protein data bank. You can find structure of mature particles, immature particles, different complexes with fab fragments of antibodies and more exotic forms. Okay, but our goal was try to perform single particle imaging experiment of inactivated tick-borne encephalitis virus at SPB as a fixed instrument of European XFL, which is most brightest free electron lasers now. But we know that a good sample is a key to success of every structural biology experiment. So special sample requirements you need to meet if you want to have good data, both for cryo-EM or single particle imaging. So homogeneity as high as possible, high concentration, absence of aggregates. It's more strictly for single particle imaging than for SPA. And especially for European XFL experiment, you need quite large amount of sample, up to 1 milliliter. And for viruses, it is not very easy to produce so large amount. And also you can to think about biosafety level, because for free electron lasers and many cryo facilities, you cannot use wild virus, so you need to use inactivated virus or attenuated strains or recombinant viruses. So to meet these requirements, we need reliable quality control. And first task was development of workflow for TBE sample quality control for SPI experiment. Here you can see the scheme of different purification protocols, which our colleagues from Chumakov Center developed. So basically, its initial sample is registered vaccine against TBE in Russia. And actually, it was 3 protocols, 2 protocols based on size exclusion chromatography, and third one based on ultracentrifugation, and we have to investigate it, which one is better. And first choice was just negative stain transmission electron microscopy. And if you're looking at the upper part of the slide, the sample from protocol 1 was not looking good. It's low sample concentration, so we discarded this step. Sample after protocol 1 was better a bit, but we saw there some kind of aggregation of broken particles, and the protocol 3 looked much better. But if you look at this image, you can see some kind of pseudo-aggregates indicated by these red lines. Why I said pseudo-aggregates? Because you don't know its effect of interaction of particles with contrasting agent or it's really aggregates. We perform CRIEM, but also we have absence of this contrasting agent, but we also have some pseudo effect of aggregates because sometimes your particles can land at different layers in the ice. Why we pay attention on aggregation? Because of the delivery system. You have to electrospray your sample to aerosol, and if you have aggregation, aggregates will clogging your capillary, which is a part of delivery system, and prevent injection. Injection is a key moment of whole experiment. If you have not good injection, you will not have data, for sure. So the sample have to be homogeneous as much as possible. And we decided to use additionally small angle scattering because this method allowed you not only detected your particle and side distributions, but also found the shape of particles and do some statistics. So you will know how much aggregates you will have a solution if you have it. And we can see also the protocol 3 produced the more nice sample. We have this narrow peak corresponding to 25 nanometers radius particles. And for protocol 1, it's not so good. And also we decided to check what's happened if we have good sample, but we store it at -80 just because for during transportation, you also can damage a bit your sample. And it's not easy, especially now. And maybe it's better to store sample at XFL just near beamline. This table is much more representative, so you can see that for protocol 3, we have 100 spheres with radius about 25 nanometers. This measurement was performed by Moore beamline and Kurchatov synchrotron just before experiment which was performed in 2019 at European XFL. And also we repeat this purification later and perform another experiment at XFL, and it's quite reproducible. The measurements in this case was performed just before beam time at Petro 3, at beamline P12. But for protocol 1, we checked it in Moore beamline, and we discovered that we have from 7 to 20% of aggregated samples, and this protocol was finally discarded. And about this experiment with sample which we stored at -80, we discovered that it was initially very good because it's the same sample which was used for first XFL experiment. But after 5 months at -80 and thawing, we discovered 10% of sphere with higher radius, about 45 nanometers, so it means aggregation was started. So additionally, before experiment at XFL, you have to check your sample at XPI laboratory. So our colleagues from XFL repeat the negative stain experiment. This is the image. Also, atomic force microscopy was done. It's confirmed the result of negative stain and dynamic light scattering. Also, it's very reliable. So we have narrow peak corresponding to 25 radius particles. Also, nanoparticle tracking analysis was performed, but here you can see a bit of aggregation, but it's really connected with method, not with sample. And the obligatory experiment, it's different mobility assay using scanning mobility particle sizer, because the conditions is absolutely the same when during injection of your sample at the beamline. So, and you check also your buffer, not only your sample. And we had two peaks here. One peak is corresponding to our virus particles. It's about 45 nanometers diameter. But we have also other peak, less strong, about 11 nanometers. And we decided this peak comes from buffer. Usually during SPI experiments, researchers prefer the volatile buffer like ammonium acetate. But when we tried to dialysis our sample, which were nitrate buffer and a bit of sodium chloride, it led to aggregation, and we decided to perform experiment just in buffer, which was used for purification in last step. So it was performed in trees and 150 millimolar sodium chloride. And finally, you can see the representative images from single particles. The particles has a bit different size, so from 40 to 54 nanometers diameter. And here is a distribution of particle sizes estimated by fitting spherical form factor to the radial profiles of diffraction patterns. But what about Creo-EM? This question was raised by our referees when we submitted paper to the journal. And they said, okay, you said you have very good protocol, good sample, but how your Creo-EM structure can look like? So, and we collected data from sample which was produced using protocol 3, which was the best one. And the data collection was performed in Titan Cryos at Kurchatov Institute using Falcon 2 detector. And here is radially colored virion shown the direction of 5-fold symmetry and central cross-section of Creo-EM structure. And we can look at 2D classes. First class is material particles, and you can see we have more than 80% of this type of particle. We have a bit of immature particles. We have more than 10% of elongated particles with broken symmetry. We have a bit of damaged particles and half-matter particles. So the 2D class corresponding to mature particles was used for 3D classification. And here's a representative image of Creo-EM denoising and workflow of processing. And finally, we had 3.02 angstrom resolution. Here is a local resolution map and Linear plot and FSCS plot. And if you will search in PDB structure of tick-borne encephalitis, you will find two structures actually. First structure of hyperstrain was solved at 3.9 angstrom resolution in 2018. Second structure was solved recently at resolution 3.3 angstrom. It was Western type Kulat-Sala strain. And now we had third structure of Russian Sofrin strain. And let me summarize the results. So developed pipeline of tick-borne encephalitis sample, encephalitis virus sample quality control was very useful both for SPI and SPA experiment reconstruction. So in using combination of different methods is a key to success. And now we have the best resolution for tick-borne encephalitis virus. And due to developed workflow, we collect diffraction data set from TBEV at SPB's effects instrument of European XFL, and diffraction data from 50 nanometer enveloped virus were obtained at this instrument for first time and shown the possibility of instrument, and our success investigation pinpoint importance of using fresh sample for injection. And develop purification and quality assessment pipeline may be useful for the preparation of other challenging viruses sample for single particle imaging and different free electron lasers. And finally, I want to say thank you all people who participate in experiments. So first of all, I want to say thank you to my colleagues from Kurchatov Institute, they are co-authors of the paper, Evgeny Pichkur, Petr Konerev, George Peters, and Mikhail Kovalchuk. Also, I want to say thank you to people from Resource Center Nanozond, which care about time crisis. Colleagues from Chumakov Center, Mikhail Varovich, who has performed this development of purification protocols. Also to Alexey Yegorov, Dmitry Solodkin, Aidarish Mohametov, and also Evgeny Khvatov, Alla Ivanova, Ksenia Tuchinskaya, Olga Konishenko, and Anton Fedorov. And special thanks to Philip Maya from Uppsala University, who was PI of both experiments at XFL and also organized preliminary injection testing in Uppsala in laboratory. And also colleagues from European XFL, Ekaterina Raut, Robin Schubert, Johan Beletsky, Richard Bean, Igor Sobolev, Adrian Mankusa, Mikhail Rychov, and Sergei Molodtsov. And of course, foundations, Russian Foundation for Basic Research and Russian Ministry of Science and Education.
Okay,
Thank you
very much.
for
Very interesting talk. Any questions? So, yes.
Thank you for presenting these excellent results. I have a question about the cryo-EM reconstruction, specifically the heterogeneity analysis. I mean, you told that you applied the two-dimensional classification to distinguish between different kinds of particles. But actually, this type of virus can be a little bit squeezed in one direction. For example, it has some flexibility, which leads to the wrong three-dimensional reconstruction when applying icosahedral symmetry. Did you encounter this kind of a behavior in this virus? And if so, how did you overcome it? Thanks.
This 2D class, which was used for 3D deconstruction, was really uniform. So this is really good sample. This is virus, which is not us. So this is really good sample for this reconstruction, because we try to work with other flavi. It's the behavior is not so good. So.
Thanks.
And the main idea to get good reconstruction was to get as pure sample as possible, right?
Yeah, but this inactivation surprisingly also helped a bit. So it seems to us, it's the virus, it's a bit, I don't know how to say, harder, maybe. So it's make the particles more, yes, this cross-linking helps somehow.
Okay, thank you. Any other questions? Yes.
Thank you very much for the nice talk. As DBIF is an enveloped virus, did you estimate whether the lipids or membrane stay intact?
Sorry, no, we didn't estimate. No, it was not our goal. By our methods, we cannot see this lipid membrane.
Okay, thank you very much.
And do you see membrane, transmembrane parts of the envelope?
When we tested first sample not very high purified, we saw the membrane separately on the images. So when the particle broken, the membrane come out, then can form the bigger, I don't know, empty spheres or something like that, and you can see it.
Okay, thank you. So right now, thank you again for your talk. And we have a coffee break now. Please come back after coffee break. We have 2 wonderful talks after that.
Спасибо. Ну, слышно. Okay, I'm sorry, let's start our session. Некому мне представить. Так, ребят, пойдемте-ка слушать мой доклад. Ходи. Ольга Сергеевна, пора начинать, наверное.
Коллеги, заходим. Okay, colleagues, we have our last part of the session, and today we have our speaker, Zakhar Shenkarev, who will speak about SARS-CoV-2 variants and antibodies. So let's start our session.
Okay, today I speak about antibodies that neutralize coronavirus and can be used in clinic to combat this disease. What we know about pandemic of SARS-CoV-2 is similar like previous pandemic of SARS-CoV. It's induced by coronavirus, but it's have much larger spread, about 7 million of deaths worldwide. Almost all countries are influenced by this epidemic, and more than 700 million of cases. Similarly, like previous coronaviruses, this virus uses a special protein on the human cells. It's angiotensin converting enzyme 2 on the human cells is also present on the blood cells. And similarly, like previous viruses, it requires some cellular protease to become activated. Some bonds should be cleaved by proteases to activate virus. And also similar like previous cases, some antibodies from convalescent serum could be used to treat the infection immediately after disease start and make the patient feel significantly better directly in short time. Here, the structure of spike protein or S protein of coronavirus, you could see that it's expressed as one long polypeptide chain, that it contains 2 subunits with the cleavage sites S1, S2. There is 2 subunits from center of the spike protein, the trimer of these S2 subunits from the rod, on which is assembled 3 N-terminal subunits, S1 subunits, that contain 3 domains. And it's N-terminal domain, receptor binding domain, or RBD, and C-terminal domain that can be subdivided in 2 subdomains. So they have such trimeric assembly with 3 S2 subunits and 3 S1 subunits. These assemblies are inserted in the virus membrane, but on the Crioelектron microscopy maps, the membrane part usually invisible. There are dedicated studies by NMR about structure of this trimeric membrane part. How this protein work? Indeed, the receptor binding domain, or RBD, could be in two different conformations, and RBD down, it's hidden from the outside, and in the BD up, it can attack the target receptor. Then the virus cell, virus approach to the target cell, the RBD is open and bind to the target receptor, and this binding starts a number of reactions. First of all, some proteins cleave the S2 subunits on additional cleavage site S2 prime, and after that fusion peptide is released, and like jackknife, it's pushed to the target cell membrane, and thus the S2 subunits join together both membranes and induce their fusion. S1 subunits dissociate at this moment. Finally, this led to the post-fusion structure that is linear, contain only trimer of S2 subunits. There are a lot of different structures of spike protein or individual RBD in complex with its target, with full receptor RCA2, or with individual peptidized domain of RCA, the target of RBD. The wild-type virus in the structure of S protein from wild-type virus usually have all RBD in closed conformation. Probably this protects the RBD from attack of human immune system, but it's slow the virulence of the spread of the virus. And in modern version of virus, in all versions after alpha version of virus, this contain this mutation that induce dynamic of the RBD, and this S protein from this modern type of viruses contain usually one erected RBD or two erected RBD. Thus one RBD in down conformation and two in up conformation, or vice versa. Here the structure of RBD, the green show so-called RBM, receptor binding module, is the place where the target receptor is bind, and the point show different spheres, show the points their antibodies to the virus are bind. The red spheres indicate their neutralizing antibodies are bind. So you see that antibodies bind to RBM can neutralize the virus and and stop its spread. Indeed, if you use only one antibody to treat infection, very frequently the mutant virus is found in the culture after passages, and they found new virus that escape this treatment. So usually antibodies used as a cocktail of two different antibodies that bind to different sites on this RBD. For example, this cocktail of two antibodies commercial, this bound to RBD in two different places near the receptor binding module. They investigate this antibody, Regen 10987. This bound on the periphery of RBM, and this cocktail was used to treat the Trump when he obtained the coronavirus infections in the USA in 2020, 21. So they try to do structure of our spike protein. They use delta variant of spike protein, the FIB fragment from this commercial antibody, but this was not commercial. It's prepared in lab by recombinant expression. And they obtain structure with approximately overall good resolution, 2.5 angstrom, but this resolution was good only in S2 part of the spike protein. The periphery was very badly resolved. The RBD and bound FIB fragment was also not very well resolved. Therefore, they do 3 steps of focused refinement of individual RBD with bound FIB fragments, and obtain 3 additional structure of these complexes with resolution 3.2, 3.4 angstroms. Here the overall model is shown. You could see the trimeric S protein with 2 RBD in up conformation and 1 RBD in down conformation, and 3 FIB fragments that attach to this RBD. The FIB fragments that I'm wrong, because only one RBD in up conformation in the FIB3 is attached to the side of this assembly, and two RBD in down conformation with FIB attached to the top of the assembly. There is a similar attachment side, but because RBD have different conformations, different position, the FIB fragment is attached from different sides of the whole assembly. Here, the one of these structures, you could see that indeed FIB fragment interacts with both light and heavy chain to the part of the RBM on the side of this part. How these antibodies, if you compare with previous structure, it was determined with resolution 3.9 angstrom. They see some difference here that can explain, probably think that this difference due to fact that we have better resolution in this part of the molecule. We have some difference on the interface between RBD and here in the FIB fragment. How this antibody is work? Here the map structures of complexes of RBD with peptidase domain of ACE2 and our complex of RBD with FIB fragment. They see the clash between the structures of peptidase domain and FIB fragment. That means that if the FIB fragment is bound to the RBD, it prevents the binding of RBD to the angiotensin conversion enzyme number 2. Thus, our antibody should be protective. It should neutralize virus and stop its distribution in the body. But different sides of interaction between FIB fragment and RBD have different dynamics. And to analyze these dynamics, we use classification, 3D classification of all our data. We made 6 structural classes and see how much of density are presented for each FIB fragment in each class. The FIB number 1, that is bound to RBD number 1, this one, is observed, it's fully observed in 5 classes from 6. That means that this FIB fragment have lesser dynamic. The FIB fragment number 2 is observed fully in 3 classes, but in other 3 classes they have low density for this FIB fragment. That means that this FIB fragment and RBD to which it connects have more dynamic. But the 3rd FIB fragment was observed fully only in 1 class. The other class demonstrate very vanishing density for this FIB fragment, which means what FIB fragment number 3 is much dynamically and is more dynamic than other FIB fragments. To illustrate this dynamic mobility, they use 3D viability analysis was implemented in CrioSpark, and we obtained 4 major modes of motions in our complex. And you could see what first mode of complex, it's originated with moving of FIB2 fragment with RBD2 domain, but other 2 domains are intact. But the second complex indeed includes the large movement of 3rd FIB fragment and 3rd RBD in concerted movement of 2 and 1 FIB fragment and RBD. You also see the concerted motions of these large guys is the N-terminal domains. They usually determined with low resolution because they are mobile, and they also moved synchronously with other motions. But if we look on the 2 last modes of motions, they observe that these modes are connected to the movement of FIB3 fragment. And all other 2 fragments move concertedly but in different directions. So from 4 modes of motions, 3 modes are involved motions of FIB3, and only one mode involves motions of FIB1 domain. And our structure permits to answer on some questions that could be addressed about active spike protein. For example, literature frequently discussed that interaction between RBD down domain and RBD up domain, then these interactions should support the movement of this domain to an up position to the activation of spike protein. Indeed, they observed the interaction of some loop from the RBM of down domain to the some path, from the RBM, from the RBD of up domain. Here, these interactions shown in details. Then take our FIB fragment and analyze its binding to the RBD domains from different viruses. They analyzed it using thermophoresis. They observed what they have very strong binding to in nanomolar scale to Utah variant of wild type variant of coronavirus, to Delta variant of coronavirus, but have almost no binding to Omicron variant of coronavirus. Why Omicron variant escape the neutralization by this antibody? We try to answer on this question. The Omicron variant is different from Delta variant, but 15 mutations in the RBD domain. You could see that all these mutations are located close to the RBM part of the domain. And in principle, all these mutations could influence the binding to antibodies or the binding to the target enzyme on the target cell. But on the interface of binding between our antibody and RBD, there are only 4 mutations are found, these mutations here. But if they look on the structure directly, they observe that only two of these mutations involve residues that directly interact with the antibody. First residue is glycine 446. It's formed packing context with asparagine 57. And in mutated variant, in the Omicron variant, this residue is substituted to serine, and probably that led to van der Waals repulsion between antibody and RBD. And second place is the asparagine 440 that is replaced in the Omicron variant by lysine, and in this case should be electrostatic repulsion between lysine from heavy chain of antibody. To analyze this difference, they use molecular dynamics and calculation of free energy changes by computer calculations. If we run molecular dynamics calculations for complexes of Omicron and Delta variants with antibodies, they observe that Delta variants, the complex with Delta variants are stabilized by average by 7 hydrogen bonds, but complex with Omicron variants are stabilized at average by 3 hydrogen bonds. That indeed confirm that the complex with Omicron variant have low stability. But if they calculate the delta G variant, delta G difference using umbrella sampling, they observe several strange observations. First of all, they use very large difference between individual run of calculation. That indicate that the structure of the complex of antibody and RBD is not stable, it's moved, and the calculated energy depends from the structure what we used for the start of these calculations. Therefore, we are forced to take several rounds of calculation and calculate average with some errors. They observe significant statistical difference between Delta and Omicron variant energy, but also they observed that this, the minimum energy for the N440K mutated RBD. The energy of interaction in this case was lower than the energy of interaction in Omicron variant. The other mutations doesn't have so large impact on the complex. To explain why this mutation is so drastic, why it disrupts the complex, they analyze the contact in our long molecular dynamic trajectory and see what if they compare the structures of delta with this individual mutant, point mutant, they observe a lower number of complexes in the part of this mutation, in the part of Glasson mutations, and in this part of the structure. But if we go to the, if we compare Omicron and Delta variant, they observed similar decrease in contacts here, but some increase in contact in the vicinity of other two mutations. Thus, we can conclude that these two mutations are partially compensate the negative effect induced by mutations of asparagine 440, and thus the Omicron variant is marginally stable but not completely unstable like variant that contain this single mutation. Is the first part of story what I would like to talk today, but the second part of story was about the completely different antibody, about nano-antibody. The nano-antibody is produced recombinantly, but is originated from llama immunoglobulins, and it contained only one immunoglobulin domain of heavy chain, and this very small domain, about 15 kilodaltons, but this domain contains all needed things to bind tightly to some molecules, in our case to RBD of coronavirus. So here the comparison of particles what we use. In first part of the talk we use FIB fragments with mass about 50 kilodaltons, and now they use a small VHH fragment with mass about 15 kilodaltons. This is the structure approximately of this type of antibodies. It's contained only one immunoglobulin domain with these loops that usually binds to the target receptor or target protein. And the antibody, nano-antibody what we use binds to delta variants of coronavirus and neutralize it. And we try to describe how it works, where it's bind, and what the structure of this complex. Initially, they took delta variant of coronavirus and made the structure of complex, but this structure shown here, in the structure is fitted by the structure from PDB of delta spike variants with 2 RBD up and 1 RBD down. You see that it's perfectly fit, but there are some additional densities that corresponds to the bound antibodies. And here almost 100 particles in this structure. But if this see the resolution of this structure, we observe that resolution in the core part of its two subunits is quite good, but the resolution on periphery is bad, and especially it's bad on the RBD contacts our nano-antibodies. It's also bad in the N-terminal domains, but at normal, it's usually found in the structures of spike protein. Here, they also guys with low resolution, it's glycosylation. They also have a small resolution, but they need some trick to enhance the resolution on the binding interface to see how these interfaces work, how the antibody binds to our RBD domain. They try to divide our data to the several structures to find if they have some structural different conformation in our data set. Indeed, we found two structures, each with resolution about 3.5 angstroms. They contain about 40,000 of particles and 30,000 of particles, and each represent individual structure with quite good resolution. If they compare these structures, they observe that these structures are different by the number and size of their antibodies bound to RBD domains. For example, here shown the fragment of the structure with RBD down. On the first structure, they see only one antibody that is bound to the site of this RBD. But on other structure, they see two antibodies that is bound both on the site of RBD and on top of this domain. If we move to the next domain, next RBD, it's RBD up number 2. They observe what here, what we see in RBD up, both sides are occupied. One antibody bounds from the side, other antibody bounds from the top. But in this case, they observe that RBD down have no binding to the site. That means that here is not additional antibody is not fit to this cavity. But another structure, they see that this antibody in this cavity indeed bound to the RBD in down state, but RBD in up state contain only one antibody that bound to the top of this domain. If they look on last RBD, they observe in both cases similar binding of two antibodies, one bound to the site and one bound to the top of domain. So in our structures, they have 5 antibodies in total bound to our 3 RBD domains, and 6th antibody does not fit to the structure. There's no room to fit the 6th antibody. And the way how the 5th antibody bounds to the RBD in down states or RBD in up states is determining the difference between our set of structures. If we look on RBD domain and observe they bound to the antibodies, they see that one of the binding interface in the top, it nicely corresponds to the RBM. Thus, the binding of non-antibody to this site indeed should prevent the attack of coronavirus, should inactivate it. But the second interface, it's located on periphery, it involves different parts of the secondary structure, and this interface is probably not needed for blocking of coronavirus. But our data doesn't permit, at present state, doesn't permit to analyze the structure of antibody. They don't know how an antibody binds to the RBD because they have low resolution in the antibody of the antibody fragment. Probably they need Fox refinement to resolve this task and to make our structures, our 2 structures more clear, to observe indeed how this antibody work. Here I would like to acknowledge the people who participate in this work. It's different institutes what engaged in the Курчатов Institute, НИИ физико-химической медицины, Shenzhen Group University, and our IBH run. Thank you.
Okay, thank you very much. Very good talk. Thank you. So, any questions from the audience? Yes.
Thank you for your presentation. Can you say something about difference between binding Fab fragment of antibody and full antibody with RBD fragment of spike protein?
It's difficult to discuss this different on the basis of Cryo-EM data, because for structural investigations usually used Fab fragments. If we used full antibody, it's very flexible and they have no resolution for the...
But it's known that full antibody, binding full antibody.
I see only one structure, the full antibody was investigated, and they have a black hole in the antibody there.
But maybe not by cryomicroscopy, but
X-ray?
You see it by just binding between...
They did not measure it. No, okay. I can't comment.
Okay, any other questions? No questions? Thank you very much. Okay, and the last speaker in this conference is Tatiana Stanishniva-Konovalova. She's a senior researcher from our department of bioengineering. And she is my former PhD student, but now she's already almost on her own. And she will talk about viral and bacterial chaperonins.
Hello, dear colleagues. You can hear me well, right? So I'm very excited to be here, and as a member of the organizing committee, I would... like that. So as a member of the organizing committee, I would also like to thank all the lecturers that gave us their wonderful presentations. I'm very flattered to be on this list. And my my talk today will be about the so-called chaperonins protein family. I want to tell a story about how RICCEM not only gave us the new structures of the brand new members of this family, but also how it allowed us to get new structures of the very well-known, basically the textbook example of chaperonins. First, let's take a look at the cell. Always a nice thing to do. So this is the scheme of generalized cell. And this is all about the protein folding. So we all know that it is not enough to just synthesize a new protein chain. You also have to fold it into the native structure for the protein to perform its function. And we know that the protein folding has been a long-standing problem. And Professor Shaitan gave us a lecture 2 days ago on that. But the current understanding is that the protein does not have to solve this problem on its own. It has many assistants in a cell. So they are called chaperones. Here on this picture, you can see many members of them. The chaperonins is just one of the many subfamilies of these chaperones. They are also called HSP60 for heat shock proteins 60. Let's take a look at the ribosome. Here is the native newly synthesized chain. And you can see that this yet unfolded chain can be caught by several members, and in some cases they help to fold this protein, in some cases they take it out of the cell. The HSP60 family, which we are interested in, it acts on different stages of this process. For instance, it can help to fold the protein into the native structure. It also can help the partially unfolded protein. If something goes wrong and the protein becomes partially unfolded, they help to prevent aggregation of such proteins and the development of some bad conditions such as amyloidogenic diseases. Of course, these proteins were united in the same group based on their sequence similarity, but also because they have very typical, very recognizable structure. I will show it on the next slide. I think that you have all heard about Grow-S bacterial complex. It actually belongs to the chaperonins of the first group, together with the variants from mitochondria and chloroplasts. There is also a second group of chaperonins. These are variants from eukaryotic cytosol and from archaea. But I want to focus a little bit more on the Grow-S complex because this will be the first part of my talk. This is the textbook example that has been studied for several decades. I think that the high-resolution structures were already available in the 90s, achieved by crystallography. This complex is composed of 14 identical subunits. Let's take a look here at the upper left side of this picture. So these 14 subunits are arranged as 2 rings. Each ring contains 7 identical subunits, quite simple. I will be calling this kind of view a side view of the molecule, and this one will be the top view of the molecule, like rotated 90 degrees. So you can see here that this is quite a symmetrical structure. It is always nice to work in cryo-EM with structures that have symmetry. Because this complex, in this complex, the subunits are in the same conformation in its upper form. So how does this complex assist protein folding? Well, the basic idea is also quite simple. Each ring possesses the central cavity, and inside this cavity there is a hydrophobic lining. And when the protein is partially unfolded, it has hydrophobic residues exposed on the surface, which is recognized by this hydrophobic lining, and that is how the protein gets trapped inside this cavity. Then the co-chaperone, which is GROW-ES and is also a ring composed of 7 subunits. It acts as a head, which is yellow here. It closes the chamber with the protein trapped inside. And here, in this closed environment, the protein has a chance to fold into its native structure. Importantly, these conformational changes, these switches, they occur in an ATP-dependent manner. So for all the subunits to change their conformation and to catch this yellow head, you need to give them, each subunit, an ATP molecule. This ATP binding itself triggers the rearrangements, and while the ATP is not yet hydrolyzed, the protein has time to fold. And then, when the ATP molecules leave, the complex loses GROW-ES and the protein is released into the environment. The chaperonins of the second group work similarly. Structures are similar with several differences, such as they don't have this GROW-ES head. They work without co-chaperone. They also have different subunits, so they have they are less symmetrical by their nature. But we are now not going into the details about them. I want to show a little bit more about the tell a little bit more about each individual subunits and how it is arranged. So each subunit in GROW-ES complex contains 3 domains. The violet one is the apical domain. It is responsible for interactions with the protein and with GROW-ES. The gray one is equatorial domain, and it is responsible for holding this whole complex together and also for ATP binding in hydrolysis. And the intermediate domain is a connection between the equatorial and the apical ones. Importantly, between these domains, there are so-called hinge points made of proline histidine residues, and they allow these domains to move as rigid bodies relative to each other. And that is how you get the elevation of the apical domain and how you get the conformation that is suitable for GROW-ES binding. So this was a basic introduction. This was all known many years. In fact, this picture is already slightly outdated because nothing is said here about the existence of the phage chaperonins, and they were already found, at least they were found in the genomes as the sequences and GROW-like sequences, but I will talk about them in the next part of my talk. Now I want just to talk more about GROW-ES. The GROW-ES functional cycle, again, this is a picture from the same review, and it is kind of simplified because there are new data that kind of will make it outdated. But just to make it simple, I will focus on this one. Again, the idea is quite simple. Here you have this empty, let's say empty complex. Here you have the unfolded protein and green ATP molecule. So green ATP molecule binds to one of the rings, and the unfolded protein binds to the apical domains. Then what happens? The apical domains elevate. They catch the violet GROW-ES head. The protein becomes trapped into in the folding chamber. Then the ATP slowly, or not so slowly, depends, hydrolyzes, and you have now ADP molecules, and now you have the second ring beginning to do its job, so to bind another molecule of substrate protein and another 7 ATP molecules. Here you have what they called bullet type of complex, because it is like shaped like a bullet, basically. And you only have bullets in this cycle, so when the second ring binds ATP, it kind of triggers conformational, creates conformational tension in the equatorial part of the complex, and this causes ADP molecules being released and the GROW-ES also to be released. So now you have another bullet, just if you rotate it 180 degrees, you you are at the beginning of the cycle. So this is the bullet cycle. The football cycle is called so because there is another shape that looks like the American football. Basically, the idea is that both rings work simultaneously. So they bind ATP simultaneously, they bind protein simultaneously, and that's how you get this football shapes. X-ray has obtained structures supporting both kind of cycles. Now there is also data on about the fact that rings can separate. There was a paper published in 2021, I guess, that in the course of the cycle, the rings get separated, and there can even be an exchange between different GROS complexes with these rings. But let's focus on bullets and footballs for now. Actually, we started studying GROL because we had a project on interaction between prion protein and chaperoneans. And at first we decided that we need to look at just chaperoneans before we study the interactions with the other proteins and just to get a reference structures. So the first reference structure was empty GROL. It was like fine. It was nothing new about it. And then we wanted to do a GROS structure and compare it to the available structures in the PDB. So I was sure that at this time, and it was 2019, 2020, there must be high-resolution cryo-EM structures of GROS complex from cryo-EM. But I was surprised that there were none of them. So all of the high-resolution structures were obtained by X-ray. Here in the table, I summarize what we had at the beginning of 2020. So here is the PDB. Oops, I want to go back. Spoiler. Here's the method of structure determination. Here is the form. So just to remind you, bullet is this one, football, or this one is football, and this one is bullet-shaped complex. So while X-ray gave us both footballs and bullets, Cryolextron microscopy only gave us bullets, which is probably an argument in the fact of bullets being a more physiologically relevant type of complex. Interestingly here, if we look at the ligands in the ring, if we are talking about the football shape, we have nucleotides in both rings. Well, that makes sense because to get this GROS head, we had to change the conformation, so we had to bind the nucleotide. And if we look at the bullet complexes, the nucleotides were only present in the GROS bound ring, which again makes sense. But if we look at cryo-EM, this was surprising for me because the best resolution was 7.7 angstrom. So these were structures published in like Cell and top journals, but they were published in the beginning of 2000, so the resolution was just, it was not possible to get high resolution structure. And I decided that, we decided that we need to get one structure because it, even if it's just exactly the same as high resolution X-ray structure, still it will be the first cryo-EM high resolution structure of GROS. And at that point I became kind of anxious about it because this idea is very simple, and I think that it must be in someone else's head and someone else is doing exactly the same thing, and we won't be the first to get high resolution structure, but we actually managed to publish these structures. We got even two structures at 3.4 angstrom resolution. They are deposited in protein data bank. They were both bullet-shaped complexes, and what was very unexpected, they contained densities for nucleotides in both rings. This is again unexpected because if this ring does not have GROS, why would it contain nucleotide? Why didn't it leave? Why didn't it change its conformation for GROS to bind? We also kind of speculated that this nucleotide is ADP because the resolution of the equatorial part of the complex is usually the highest one because this is the most stable part of the complex. And we believe that this is ADP molecule. So we have two structures with the same nucleotide occupancy in two rings, but they were conformationally different. here are these two structures. They were quite similar in the part with the GROW-S on top, but they were quite different in the apical domains of the second ring. So here is the difference. We even turned them like differently. We published this paper in 2021 in Scientific Reports, and we kind of go into the details about which part of functional cycle are these structures looking like. So if you want to go into the details, you can read this paper. I just want to give the general idea that it is still possible to get new results on the basically textbook example of a protein. Here is these structures with colored according to local resolution, and as I said, the best resolution is always in the equatorial part of the complex, and the apical domains are blue. They are the most mobile parts, and they always have a lower resolution. In the second part of my talk, I want to get from this well-known chaperonin to the yet almost unknown area. These are the viral chaperonins. And just, I think I will immediately give you the structure that we obtained in 2020. This was the structure of chaperonin from phage OBP, from Pseudomonas fluorescens, and I will call it OBP chaperonin. From the previous experiments, from preliminary experiments, including negatively stained microscopy and biochemical experiments, we already knew that this won't be a tetradecamer, so it's not like GROW-L, it is a single ring. And from all of the experiments, it was a single ring composed of 7 subunits. But they were all the same subunits from the sequence. But unexpectedly, when we tried to make the reconstruction using C7 symmetry, the results didn't make sense. We didn't have enough density in the apical domains. And at first I thought this is like me, because I was just starting to get familiar with data processing in cryo-EM, and I thought maybe that's just, I cannot get this right. But eventually it turns out to be that the problem is the chaperonin, that it's just asymmetrical in its nature, although all of the subunits are the same. This pattern of subunits is visible at the level of the apical domains. So here, if we do slide, this is the side view. Yes, this is the top view of the molecule. If we get sliced through the equatorial domains, you see this like typical for a single ring of GROW-L, C7 symmetrical pattern. But if you move up and make a slice through the apical domains, you see something very unusual. So we were able to distinguish 3 conformations that we termed A type of conformation and 3 B conformation. And these A, B subunits, they have a set of solid bridges between them, so they are united in pairs. So we have, out of these 7 subunits, we have 3 pairs of subunits, and 1 subunit is left unpaired. And this subunit is not quite visible in our reconstruction because of its higher mobility. So I remember when I first tried to feed the atomic model into this into a single subunit, it was okay at the level of the equatorial domains. It didn't fit at all at the upper part of the complex. I was trying to move, of course the model was based on GROW-L because the sequence is similar to GROW-L, and I was trying to move around like helices to make it fit better, and then Professor Sokolova was coming by and she said something like, just break its neck and turn its head, and I like broke its neck, turned the head, like completely turned the head, and it fit well. So it has a very strong conformational differences to GROW-L. And we did not know at first why, but quite soon the explanation from the bioinformatical approach came. The thing is that at the level of the top hinge points, remember I said that between the apical and the intermediate domain there is a hinge point that allows the rotation of the domains as rigid bodies relative to each other. But in case of this OBP chaperonin, in its sequence, the proline residues were substituted to larger glutamine residues, so this apical domain was fixed relative to the intermediate one, there was no rotation possible. And it was fixed in a quite unusual, comparing to Grouil, position. So this rotation around this point was impossible, but the lower hinge was working, and this was responsible for the existence of two conformations, A and B. So what is surprising here that in the absence of any nucleotides or substrate proteins, we still have the existence of two different conformations. And it looks like the 7th subunit is switching between those two conformations, and that is why it's kind of invisible on the reconstruction. And of course, I mean that the 7th subunit is not always like the same one. It's just that I think the pairs can switch. So this subunit can form a pair with this one. They will change their conformation, and then another one becomes like free and less visible. This is the comparison of those two A and B conformation. Again, this was published in 2020. And again, I was kind of nervous when we obtained these results, because I thought that, well, this is very unusual, this is very new, we have to publish it, because otherwise someone will do it first, which is, well, a strange idea, because this is not such a hot topic of research, this viral superonions, not so many research groups work immediately on that, but I had this idea that we need to publish it as soon as possible, so we published it eventually, and the same year, the paper about another viral superonion was published, and they observed the very similar pattern of alternating subunits, just like exactly as we observed, but since we did it first, they had to fairly state that this pattern was first described for the OBP chaperonians and cite our article, which was, I think it was nice. So we also wanted to study how nucleotides affect the structure. So we made structures with addition of non-hydrolyzable ATP analogs and with addition of ADP. What we observed is that addition of nucleotides does not give us any dramatically new conformation, but it kind of enables switching between A and B conformation, and the whole complex becomes more mobile and flexible. So on the reconstructions with nucleotide-bound chaperonians, we only see one pair of subunits, the same A and B, but just one pair, and all other subunits kind of disappear because they become more flexible. So how is this related to the function of the chaperonion? It's just, we don't know that yet. This is the topic for further research. We also, so there were two chaperonions. One was studied by us, this OBP chaperonion. The second one from the phage EL chaperonion, and they had this pattern of alternating subunits. So you think that, okay, so this is the typical feature of the viral chaperonions, but no. This third viral chaperonion that was also studied in our group is from phage AR9, and this one actually does look more like a single ring of Grouil. It does not have this pattern of alternating subunits. It's kind of very C7 symmetrical structure. It does not have this breakage of the top hinge, so it looks like basically a single ring of Grouil. Although using Cryo-EM, we were able to observe some sort of heterogeneity within the sample, and we described four types of subunits for subunit conformation. Again, here we are talking about the apostate, so without any addition of any nucleotides. You can see here that they are mostly different in the positions of the apical domain and intermediate one. And again, it's just a topic for further research to study how this flexibility of apical domains is related to functions of chaperonions. Here is again the comparison of AR9 and OBP. So I think the KL helices, so here they face downwards. These are helices from the apical domains, and here they face like sideways, just like in Grouil. We also described the set of salt bridges that are formed between subunits. But I think, again, I will not go into the details. I just want to give the general idea of them. And to summarize, here is the comparison of Grow-EL and 3 viral chaperones with known structures. So there are just yet 3 of them. Actually, we cannot even decide to put them into a new, let's say, 3rd group of chaperones because they are quite different from each other. And basically, they are as different from each other as they are from Grow-EL. So maybe we should put each of them into a single group or like find more representatives. We don't know that yet. So this is also, I think, very interesting area of research. Here is the double-ringed Grow-EL. Here is our single-ring chaperones, OBP and AR9. And here is the double-ring EL. I didn't mention that this EL chaperoning with alternating subunit conformation, it was purified sometimes as a single ring, sometimes as double ring. Here, the apostate is double ring. So you kind of see that, well, although this is a double ringed structure and this one is single ring, but they have this similarity in the different subunit conformations. But again, the double and single ring is a big difference. So, like, cannot put them together. And these are single ring chaperones, but this one does not have this pattern of alternating subunits. So this is, I'm just showing you that these viral chaperones are very new topic of research. And I think that in a few years, I hope we'll get more information, including structural information about them, because as far as I know, this is still ongoing research in our laboratories. At the end of my talk, I want to acknowledge my colleagues. First, Evgeny Vichkur, who played a major role in obtaining those structures. All of them were, all the data sets were recorded in Kurchatov Institute, and Evgeny Vichkur did the data processing and up to the reconstruction and model building. Also, I want to thank my colleagues, Professor Sokolova and Andrei Moisenikov, for doing preliminary studies, so we did not have to screen samples on cryos every time. We could do some preliminary screening on using MSU facilities. Also, the group of Professor Muranets, responsible for the biochemical studies, the protein purification, the bioinformatical analysis, and also our student Ekaterina Maslova, and the support from Russian Science Foundation. This project has successfully ended last year. But as I said, the viral chaperones is an ongoing research, and I hope that on the next RICCEM conference we'll hear more about them. So thank you for your attention.
Okay, very nice talk. Thank you very much. So, any questions? Questions? All are collaborators present, right? So they know this work very well. Maybe some other persons? Okay, everything is clear, good. So this was last presentation in our conference 2023. So I congratulate you that we are finishing our conference, and I would like to ask to set up the next presentation. So now we have some good part, I think, because we need to conclude. We need to conclude our conference. And just a second. And we wanted to give some presents to our winners. Yes, so this now finally is my presentation, right? So, and I think all I can tell is that we did it. We wanted to do a 4th Russian International Conference on Cryo-Electron Microscopy. And despite all problems and that we actually losing a lot of funding from Thermo Fisher, who doesn't wanted to give us any money, we actually found other sponsors, and we really thankful to Helicon, who was a very good sponsor. So they sponsored our coffee breaks and our lunches, and they also sponsored the presents to our winners. And winners are special thing, right? So, and also, you see, we translated, so we did our conference in combined format. It's actually pretty good because some people were here. So I myself think that seeing in person presenters and speakers, it's much more rewarding to everyone, especially to young persons. But some people who were not be able to come here or were not be able to come every day, they also can see it on Contact and YouTube. And now we wanted to start voting. So here let me announce the Best Young Speaker Award 2023. So here we see 6 young persons, 6 young speakers. And we have, can you please distribute the vote? So please give it to everyone. And I will just remind you in the meantime. So we have here Alexey Vlasov from MIPT, and he was speaking about building some vaccine, artificial vaccine. We have Tatiana Stanishneva-Konovalova, who just finished her talk on chaperonings. We have Natalia Garaeva from Kazan Federal University. She was speaking about ribosome. Yesterday, Maria Yakunina from Peter the Great, Peterburg Polytechnic University. So please mark only one person. Just, it's not, so you will vote and mark only one person. Maria Yakunina spoke about pseudonucleus formation in bacteriophages. Evgeny Pichkur from Kurchatov Institute, who was also speaking about high-resolution structure of ribosome, and Andrei Moisenko from MSU, who was speaking about ELSA cryoholder and a lot of different structures that he was solved using our electron microscopy, our microscope here. А вы не можете потом у них собрать это все? Можно ты будешь их считать? Okay, so we're voting. In the meantime, and by the end of my talk today, we should know who is the best young speaker in 2023. And let me just show you our conferences. So the first conference was in June 2017, so it was actually pretty long time ago. It was fully offline, so like at that time there's no such as terminologies of offline. All conferences were offline, right? So we were all in person and we talked to each other. So you can see that there used to be 27 speakers from 8 countries and 120 attendees. So here you can see a lot of people from, and actually FEI and Thermo Fisher was giving us some money, so we can invite several people like Alexei Sobolev or, sorry, who can, from MIPT. Okay, so actually it was pretty good start, and we wanted to continue it. So then in 2019, we already have 40 speakers from 13 countries and about 200 attendees. At that time, it was great summer, so we have very nice peonies and lilac, and we have some field trip to 86 floor of skyscraper. And actually, again, some people came from, so here this is actually Matthias Wolf who was speaking today. So there are some other people from abroad who came here in person. And then after that, unfortunately, in 2021, there was a COVID. So we made also some conference which was in combined format. So you see here, there's a lot of, but at that time we got 2 Nobel laureates. Richard Henderson was speaking online, Joachim Frank was speaking online, and actually people who was discussing, I mean, people in the field, they say, how you would able to get 2 Nobel laureates on your conference. So we were lucky, actually. And you see here, we have actually about 600 attendees, but 500 were online. And we have 44 speakers, again, 35 online, out of more than 25 countries and 150 cities. So our conference was growing, and some people came again from abroad. Some people were speaking online. And actually, Carolyn Luger, she was going to join our conference but she got some clash with different conference, and she could not communicate, she cannot connect from this conference to present on our conference. And finally, this is our conference now. So you can see, I calculated everything. So we have 260 attendees registered. Out of them, 50% were online and 130 were in person. We have actually 36 speakers, out of them 15 online, from more than 10 countries. So it's not bad. Again, despite that we don't have a big financing from Thermo Fisher, but it's very good that we got a lot of people who came here and talk to each other, talk to students. I still believe that this is one of the best events for the young people. So here are our organizers. So it was organized by Moscow-Lomonosov State University and Faculty of Biology, and also the School of Molecular and Synthetic Biology of Moscow University, and with the help of Research Center of Biotechnology, Russian Academy of Science. So here we have our speakers. We have Kurt Wurtry. Unfortunately, again, he could not come, so he sent his record. But I think it was better to sit here and to hear his presentation than to see it from somewhere else, right? We have our Yelena Arlova was present on all our conference, starting from the first one. Alexander Sobolevsky, also Konstantin Osachov, I think, joined us on the second conference, right? And Alexei Amons was first here, brave foreign speaker who just came in person from Sweden, and he presented a key lecture, and it was great. So actually we have another Mr. X, very, very secret person, and we never will tell anyone who is it. So he also presented. So we have a lot of very interesting presentations, and you can see here, this is actually the statistics from yesterday on YouTube. So you can see it was 136 persons watching our presentations during the day. Maximum at the same time, we're watching 26. So I think it's not bad. It means that people are interested in our conference. And then it was offline poster session. We used to have 20 posters, and we got actually 2 winners of the best poster contest. So the idea was that we have a committee, and committee out of 4 people. Actually, all of them are here present now. So they were going along all posters. They were speaking with the presenters, and they were signing their signature on this poster. And the best poster will be the one which will gain maximum signatures, right? So, and then, yes, here it will be announced in a couple minutes. So here I just would like to show you some reconstructions obtained, cryo-electron microscopical reconstructions obtained in Russia. So you can see we actually, 2 of them I just grabbed from today from the screen, and some of them I grabbed from the internet, from the published papers. Some are unpublished yet, but already under review, and some of them have not very high resolutions, but again, this is very important because these reconstructions were obtained using our in-house microscopes. So right now, as I say to everyone, we have 1.5 microscopes in Russia, cryo microscopes in Russia. One is the Titan Cryos in Kurchatov Institute, and we have another, maybe even 0.75% of microscope. So because our microscope is not, it's 200 kV accelerated voltage. So Andrei Moisenko did beautiful presentations 2 days ago about our microscopical facilities, but it can help us to get reconstructions of about 6 to 7 angstrom resolution, and it's beautiful microscope that can be used for checking samples and to obtain some preliminary results to see if we need to go to higher resolution microscope or we need to do something with our sample again. So also I would like to remind you that we are going to publish a special issue of Vestnik. So we invite all of you to submit the short report, but this short report is not an abstract, it's an article, but it should be about no more than 1,700 words altogether. It may include pictures, it may include references, and it should be revised. So the deadline for submitting it, it's June 30th, okay? And now, acknowledgements. So we have our organizing committee, we have our program committee, so thank you very much for all who was helping to organize this conference. because it was really a big job, and it's to all people who helped us, who helped to write to the potential speakers, to invite potential speakers, to arrange all talks, and also finally to come here and be present in this talk, ask questions. So also I would like to ask, I would like to thank student volunteers. And actually we have these 4 students, right? So if our students can come here, Daria, Naida, Alina, Ilya, Polina, who's here? Yeah, come in. Yes. So this is the... Yes. Everyone else is... Someone maybe can make a picture of them. Yes. Tanya, please. Okay. Thank you. So all other students will have their... sorry. Nikolai also. Yes. Таня, давайте. Подождите, куда он побежал? Okay. And I would like to thank also our technical support who are now helping us to translate everything, who helped us to show all conference presentations. And now it's a Best Poster Award. We actually have 2 posters. So, I don't know, you will give them. So, one is poster Sirotkin and all. Это вот этот. And another poster Kordyukova et al. And these are not just... We have some presents from Helicon.
Very unexpected.
Yes, now we can make a photograph. To send them later to Helicon. Okay, so now back to Best Young Speaker contest. Сейчас. 1, 5, 7, 8. Okay. Yes, so our best... Dan, может, ты напишешь? Best Young Speaker of Conference 2023 Russian International Cryo-Electron Microscopy Conference. So, I'm Andrei Moiseyenko. And I think it's a good choice, actually. And I did not vote. I just was speaking, so it's not... I did not make a bias. So, he also will have a present from Helicon. But we're waiting for this present. Unfortunately, it's still on its way. So, yeah, but we will give you a present later. Okay, thank you very much. So, thank you for your attention. And you see, we already have 4 conferences. They all of different color. Do you have any idea what color should be next? Okay, so the first, go back to violet. Опять violet. Okay, so if anyone wanted to say some words. Yes, okay. So, thank you very much for all of you for your attention, for your participation, active participation. Thank you for all people who were joining us online, and I'll see you in 2 years, okay?